Good day, everyone. I am Lukman Bolaji Bello, and I'll be taking you through this interesting course, Biochemistry, General and Medical 1. Course title is, the course code is BCH201. Punctuality is important and it is key. Always ensure to get all materials assigned when due. Feel free to drop any question and comment in the group, and you're also free to send me emails. The course content. This is the outline of the course: Introductory Chemistry of Amino Acids. We're going to be looking at the properties, reactions, and biological functions. Now we're going to be looking at the classification of amino acids, how amino acids classified, effect of acid and alkali on hydrolysis of nucleic acids. We're going to be looking at peptides, introductory chemistry of proteins, biological functions of proteins. We're going to be looking at the different structures of proteins. Also, we're going to be looking at the chemistry of lipids, and we're going to be looking at the chemistry of carbohydrates, as well as the method of protein isolation, purification, and identification. Amino acids are a group of organic compounds containing the amino and carboxyl functional groups. So what makes them amino acids is these two functional groups, the amino functional group and the carboxyl functional group. Without the amino functional group, you can't call it an amino acid. And the amino acids are building blocks. They are monomeric units of proteins. So proteins are formed from amino acids. You can't have proteins without amino acids. We have as many as 300 amino acids that occur in nature. However, 20 of these amino acids are called the standard amino acids. Why are they called standard amino acids? Because they occur repeatedly in protein structures. These are the amino acids found in protein structures. However, like I said, we have as many as 300 amino acids. And there are different ways of classifying amino acids. How do we classify amino acids? Amino acids can be classified based on their structure, based on chemical nature, can be classified based on polarity, based on nutritional requirement, and based on their metabolic fate. This is just an introduction. I'm going to be sending you more materials as we progress in this course. Proteins are the most abundant organic molecules of the living system. Like I said earlier, proteins are made from amino acids. So amino acids are the building blocks of proteins. Now, proteins form the fundamental basis of structure and function of life. And proteins are divided into 4 levels of organization. We have the primary structure, the secondary structure, tertiary structure, and the quaternary structure. And proteins have different uses in the body. An example is enzymes. Enzymes are mostly proteinous. These enzymes catalyze reactions, meaning they speed up the rate of reactions. Without enzymes, reactions will not take place in the body. And these enzymes are mostly proteins. This is just one of the many biological functions of proteins. Now let's look briefly into lipids. What are lipids? As a way of introduction, lipids are organic substances relatively insoluble in water, soluble in organic solvents like alcohol, like ether, etc. And they are actually or potentially related to fatty acids. And of course, they are utilized by the living cells. Now, lipids serve as the body's chief concentrated storage form of energy. The body gets a lot of energy from lipids, and the body also stores lipids for energy. Lipids can be classified into simple, complex, derived, miscellaneous lipids, and each of this class is further subdivided. Simple lipids include fats and oils. Now, nucleic acids. What are nucleic acids? Nucleic acids are the polymers of nucleotides held together by phosphate bridges. Nucleic acids are the polymers of nucleotides, meaning the way you have amino acids being the building block of proteins, nucleotides are the building blocks of nucleic acids. These nucleic acids, what do? Generally, they store and transmit genetic information. What are the types of nucleic acids we have? We have ribonucleic acid, that is RNA, and deoxyribonucleic acid, DNA. I believe we've all heard about DNA. There's no one who has not heard about DNA. DNA is a type of nucleic acid. And what does DNA do? DNA stores genetic information, while RNA transmits genetic information. So from DNA to RNA, and then from RNA to proteins. The difference between DNA and RNA is that the ribose, the sugar in DNA is deoxyribose. Deoxyribose meaning without oxygen. It lacks oxygen. The ribose there, the particular carbon atom, carbon 2, lacks oxygen, and that is why it is called deoxyribose. So deoxyribonucleic acid, DNA, and then ribonucleic acid, RNA. So the ribose in RNA has all the oxygen that ribose usually have. Nucleotides, like I said, are made up of nucleotides. Like I said, are the building blocks of nucleic acids. Now, what makes up nucleotides? Nucleotides are made up of a nitrogenous base, a pentose sugar, and a phosphate group. Without the phosphate group, it is not called a nucleotide. It is called a nucleoside without the phosphate group. But with the phosphate group, it becomes a nucleotide. The nucleotides without, like I said, without the phosphate group is a nucleoside. Now, a nucleotide is made up of nitrogenous base, pentose sugar, and a phosphate group. These nitrogenous bases are of two types. We have the purines and pyrimidines. The purines in DNA and RNA are adenine and guanine, while for pyrimidines, cytosine is found in both DNA and RNA, while thymine is found in only DNA, while uracil is in RNA. This is the structure of the nitrogenous bases. Now let's briefly look at carbohydrates. What are carbohydrates? Carbohydrates may be defined as optically active polyhydroxyaldehydes or ketones or compounds which produce them on hydrolysis. Carbohydrates are defined as polyhydroxyaldehydes or ketones. Aldehyde is a functional group, ketone is another functional group. So carbohydrates can either have the aldehyde functional group or the ketone functional group, and they are the most abundant dietary source of energy. We get about 4 calories of energy from a single gram of carbohydrates. And carbohydrates are broadly classified into 3 major groups: monosaccharides, oligosaccharides, and polysaccharides. I know you may have been taught monosaccharides, disaccharides, and polysaccharides. Disaccharide is a subclass of oligosaccharides. Mono means one, oligo means a few, poly means many. So monosaccharide, meaning just one unit, has only one saccharide unit. And what is saccharide? Saccharide means sugar. So it has only one sugar unit, while oligosaccharide, a few, maybe 2, about 2 to 9. So if it is having only 2, then it is called disaccharide. 3 trisaccharide, 4 tetrasaccharide, and so on. So disaccharide is a subclass of oligosaccharides. However, disaccharides are the most important of the oligosaccharides, and that is why they are more known than other oligosaccharides. Then we have polysaccharides. An example of monosaccharide is glucose, galactose, mannose, fructose, and others. Disaccharides, like I said, are the most important subclass of oligosaccharides. Disaccharides, we have compounds like maltose, we have sucrose, we have lactose, and the rest. Lactose is milk sugar, the sugar in milk sugar, which is just a combination of galactose and glucose. Maltose is malt sugar, it's a combination of glucose and glucose, while sucrose is table sugar, which is the combination of glucose and fructose. Then polysaccharides, examples of polysaccharides include starch, glycogen, cellulose, etc. Now, polysaccharides are compounds that are made up of many saccharides in many monosaccharides. So you have either glucose, many glucose units coming together to form a polysaccharide, or you can have different fructose units coming together to form a polysaccharide. And these polysaccharides can also be divided into two. We have homopolysaccharides and we have heteropolysaccharides. When you say homopolysaccharide, it means it is made up of only a particular a single unit, meaning it is made up of only a particular just one monosaccharide unit, like glucose. If it is having only glucose all through, even if it is 1,000 glucose, it is homopolysaccharide because it has only one type of monosaccharide unit. But if it is heteropolysaccharide, it means it has more than one type of monosaccharide unit. It may have glucose and fructose and galactose mixed together to form the polysaccharide. In that case, it is called heteropolysaccharide. Another name for heteropolysaccharide is mucopolysaccharides. You also call them, an example, of course, of mucopolysaccharide is glycosaminoglycan, which an example of GAG, glycosaminoglycan, is hyaluronic acid, chondroitin sulfate, damathan sulfate, and the rest. So like I said, this is just an introduction. When we go deeper into the course, I'm going to send you more materials. Feel free to ask questions, and you can also send me emails. Thank you.